Hi everyone, my name is Evan Jiang, a second-year medical student at the Perelman School of Medicine. And today I'm going to talk to you a little bit about pediatric epilepsy and one of the unique ways that we can approach treating this condition. So pediatric epilepsy and epilepsy in general is a neurologic disorder characterized by synchronized excessive neuronal excitation in the brain. While 70 to 80% of pediatric epilepsies spontaneously resolve, 20 to 30% of them progress to drug-resistant epilepsy. Pediatric epilepsy is particularly devastating because of the detrimental effect that recurrent seizures can have on neurodevelopment and quality of life in children. There are currently three main options for treatment of pediatric epilepsy: anti-epileptic drugs, surgical resection, and neurostimulation, the latter of which we'll be focusing more on in the remainder of this talk. One of the methods of neurostimulation that we use to treat pediatric epilepsy is called vagus nerve stimulation, or VNS. VNS is an adjunctive therapy for adults and children with drug-resistant epilepsy who are not candidates for surgical resection. VNS was first FDA approved in 1998 for drug-resistant epilepsy and treatment-resistant depression. VNS therapy is composed of 4 different parts, the first being a implantable pulse generator, the second being a spirally implanted electrode coiled around the left vagus nerve, the third being a device that programs the stimulation conditions of the subcutaneously implanted generator, and fourth is an external magnet that can be used by the patient themselves. Stimulation is typically automatically triggered by tachycardia, but the stimulation can also be initiated using the external magnet by the patient. The exact mechanism of VNS's anti-epileptic effect is not completely understood. There's multiple different hypotheses that try to account for VNS's therapeutic effect, including suppression of generalized epileptic waves and local cerebellar blood flow through modulation of the limbic system and cortex, increased GABAergic activity, norepinephrine activity, and amino acid metabolism, and finally, the hypothesis that VNS therapy causes neuroimmune modulation. We'll dive further into the hypothesis of neuroimmune modulation in the following slides. The proposed neuroinflammatory modulation in VNS involves 3 different parts. Firstly, vagal efferent fibers suppress macrophage TNF-alpha production in the GI tract through enteric neurons and the spleen via the cholinergic anti-inflammatory pathway. Furthermore, acetylcholine-sensitive T cells also become activated by vagus nerve efferent fibers and work to suppress TNF-alpha production by macrophages. Similarly, sympathetic preganglionic neurons also suppress macrophage TNF-alpha production by connecting to the splenic nerve and triggering the release of noradrenaline. Finally, vagal afferent fibers trigger HPA axis-related glucocorticoid release to mediate systemic anti-inflammatory effects. Further investigating the neuroimmune modulation hypothesis of VNS therapy, one group did a study where they looked at the gene expression patterns in peripheral blood mononuclear cells of patients after they received VNS therapy. And they found that following VNS therapy, there was a significant enrichment of genes that are associated with the negative regulation of lipid biosynthetic process, cardiac cell proliferation, positive regulation of GABA synaptic transmission, positive regulation of glutamatergic synaptic transmission, and regulation of neuron differentiation. The changes in the negative regulation of the lipid biosynthetic process is consistent with previous studies in rats showing that VNS had a significant effect on lipid composition, and the enrichment of the cardiac cell proliferation is also consistent with previous studies indicating that VNS can be used as a potential therapy for various cardiovascular diseases. Furthermore, the enrichment of synaptic transmission of GABA suggests that VNS upregulates GABA signaling in general, which is consistent with other studies that showed that the levels of GABA in the CSF of VNS patients was increased. On the other hand, downregulated genes that were found in patients receiving VNS therapy included some inflammatory markers such as interleukin-4 mediated signaling pathway, positive regulation of protein deubiquitination, B-cell homeostasis, regulation of CREB transcription factor pathway, T helper 1 cell differentiation, and insulin catabolic processes. The downregulation of the insulin catabolic process is particularly interesting because that would result in more sugar storage and less circulating blood glucose, which is similar to a ketogenic diet, which also controls blood glucose, which is the most effective and frequent non-pharmacological treatment for refractory epilepsy. Now, when they look at the functional annotation analysis of the up and down-regulated genes in the study, what they found was that in the upregulated genes, there was enriched pathways related to the extracellular matrix receptor interaction and the TGF-beta signaling pathways. The enriched TGF-beta signaling pathway has been reported to play an important role in cell growth, apoptosis, migration, and immune response. Studies have shown that dysregulation of TGF-beta can cause various diseases, including leukemia, impaired wound healing, and neurodegenerative conditions. The loss of TGF-beta signaling promotes tumorigenesis via suppression of the immune system, and the production of the TGF-beta1 isoform from immune cells has an anti-inflammatory function. On the downregulated gene side, you can see that there's a downregulation of genes related to stress, inflammatory response, and immunity. In terms of molecular function, genes encoding proteins with deoxygenase and kinase activity were also downregulated. Genes related to lipid droplets, cell junctions, and membranes also showed a decrease, and all of these data point towards the fact that VNS therapy does have a significant anti-inflammatory effect in these patients. VNS has been approved by the FDA for almost 30 years now and has been shown to reduce and improve seizure frequency over time. As you can see from the figure on the left, a study that followed 44 adults and 21 children over a long period showed that after a period of about 10 years, almost 70% of participants showed an average reduction in their seizure frequency. However, that leaves a remaining 1/3 of patients that do not respond well to VNS therapy. And for clinicians, this is an important question to ask about how we can determine a prognostic or predictive factor that can determine VNS efficacy. And in particular, for pediatric drug-resistant epilepsy, which is a very heterogeneous disorder, there are many different predisposing factors that need to be considered before trying VNS therapy. For example, pediatric drug-resistant epilepsy must consider whether they have focal or generalized disease, what the epilepsy syndrome is, the age of seizure onset and seizure frequency, and possible comorbidities such as developmental delay or intellectual disability, among other different factors that could be considered in the prognostic or predictive factors when determining VNS efficacy. Study trying to determine some of the prognostic factors for VNS therapy started with 45 drug-resistant epilepsy children who received VNS treatment at Chiu Hospital of Shandong University in China, and found that 25 out of 45 of the participants showed significantly reduced seizure frequency, and 6 of them actually achieved seizure freedom. These patients also showed an increased significant in quality of life and a reduction in seizure severity. And when they look at all of the preoperative variables and controlled, what they found was that 3 different factors were associated with a poor prognosis of VNS efficacy. Those being the major seizure types. So individuals that had generalized epilepsy tended to have a less of a therapeutic effect compared to others. Patients that had longer seizure durations and patients with comorbid intellectual disabilities also received less benefit from VNS therapy compared to others. Using the results from their study, the researchers created a nonogram model that can be used to predict VNS response in drug-resistant epilepsy children. Using the 3 prognostic factors they found previously, the presence of comorbid intellectual disability, the presence of longer seizure durations, and the presence of generalized epilepsy, they created this model that can be used by clinicians to try and determine whether or not a patient preoperatively will be responsive to VNS therapy or not. To summarize some of the things we talked about today, VNS therapy's therapeutic mechanism involves neuroimmune modulation involving the sympathetic nervous system and both vagal, afferent, and efferent fibers. VNS therapy resulted in downregulation of genes related to stress, inflammatory response, and immunity, supporting the neuroimmune modulation hypothesis. We also looked at a study that identified 3 prognostic factors: generalized seizures, longer seizure durations, and comorbid intellectual disability as poor prognostic factors for VNS efficacy in pediatric drug-resistant epilepsy. The results of that study still need to be evaluated by larger scale clinical trial data. And further work can be done to find and identify biomarkers that could predict VNS efficacy. Thank you for listening to my presentation, and I hope that this talk about VNS therapy and pediatric epilepsy garnered some interest in these topics. Thank you.